Hi, my name is Carmela Vita, and I'm a postdoc at Washington State University. I'd like to acknowledge my collaborators Dan Buckley, Sean Murphy, Phil Robertson, and my fantastic mentor Sarah Rowley, whose previous research really inspired this project. Today I'm going to talk about the effects of rainfall frequency on nitrogen fixation and switchgrass. Switchgrass is a model perennial grass, ecologically and practically, for its potential as a bioenergy crop. Here are three perennial bioenergy cropping systems in Wisconsin: switchgrass, native grass, and restored prairie. On the y-axis here, you have above ground net primary production or harvest. The green bars represent the fields that were fertilized. The orange bars are the unfertilized fields. I think what's particularly striking here is that even when we're removing large amounts of nitrogen in the form of harvest from these fields every year, that adding nitrogen fertilizer doesn't significantly increase production. In fact, if we look at the second year of switchgrass, we see that the fertilized plots actually had lower biomass than the unfertilized. What is it about perennial grasses that make them thrive in low nitrogen environments? Well, we know that they tend to be efficient with their nitrogen use. Their tissues are often lower in N content, and they have the ability to translocate nitrogen from the shoots to their roots prior to senescence. Many perennial grasses form strong relationships with arbuscular mycorrhizal fungi, which can increase plant nutrient uptake. More recently, we have also learned that perennial grasses like switchgrass have a cultivated microbiome with lots of nitrogen fixers. This heat map on your right shows switchgrass tissues, rhizosphere soil, root, stem, and leaf, and various microbial taxa, all with the ability to fix nitrogen. The red shades indicate those places where these particular fixers are more abundant. So clearly there are microbes present capable of nitrogen fixation. But how important is this process to switchgrass? A few studies have shown that 15N2 can be incorporated via nitrogen fixation into the roots and the rhizosphere soil. But they've also found that this process is highly variable and challenging to predict. Another way of describing this is that we occasionally catch episodes of nitrogen fixation, but they seem to occur on different channels at unpredictable times, and we cannot seem to locate the remote control. In our quest to discover and locate this remote control, our group has put forward a hypothesis to explain these episodes of nitrogen fixation. We think that soil wetting events may increase fixation because re-wetting a dry soil enhances microbial activity or the carbon being mineralized. Nitrogen fixation is an energetically expensive process, so this boost of available energy could really enhance conditions. Additionally, wetting the soil lowers oxygen levels, which is more favorable for nitrogen fixers. Because plants can also influence the rhizosphere soil, for example, by releasing carbon in exchange for other nutrients, we think that different stages in the plant's growing season may influence nitrogen fixation. I won't be examining plant phenology in detail during this talk, but please see Sarah Rowley's talk in this session for more about this aspect of the project. To manipulate rainfall frequency, we used rain exclusion shelters, as shown here in the lower right. We collected rainwater via these gutters that were funneled then into these irrigation tanks. This experiment was conducted on unfertilized switchgrass plots at Kellogg Biological Station in Michigan, and we replicated our 3 rainfall treatments in 4 plots, as you can see up here on the right, for a total of 12 shelters. All plots received 60 millimeters, or about 2 inches of water during mid-June to mid-July, which is the average for this time period. Constant moisture plots received 15 millimeters of water every 5 days for a total of 4 wetting events. Moderate drying plots received 30 millimeters every 10 days for 2 wetting events, and extended drying plots received 60 millimeters all at once on day 20. These stars represent sampling events, so we sampled before and after wetting, and by after wetting, we mean 4 to 8 hours after the irrigation finished. We also continued sampling as the soils were drying down after the final wetting event, so 2, 5, and 10 days later. For each sampling event, we collect a pair of soil cores per plot within 10 centimeters of a switchgrass crown, and they're 10 centimeters in depth. To measure fixation, we use a 3-day 15N2 lab incorporation assay. So we fill the headspace of these cores with 78% nitrogen gas, just like in the atmosphere, 10% hydrogen, and 12% oxygen. We used hydrogen, which many microbial organisms can use as an electron donor, as our energy source. That's in contrast to the more traditional source of glucose or a carbon cocktail, but that involves wetting the cores, which isn't ideal for our study. So here's the first of the 3 slides where I will be showing you the response of nitrogen fixation to different wetting regimes. Nitrogen fixation is on the y-axis, and these are the constant moisture plots that received 15 millimeters of rainfall every 5 days, as marked by these thin blue arrows. All 4 plots are represented by either a circle, triangle, upside down triangle, or a square. The X marks the mean for a sampling event, and the lines connect the mean so you can see the general trend. And what we found is that these small wetting events, at least early on, could increase fixation, especially if we accidentally doubled the amount of water. Here you see a huge increase in fixation. And that this response to wetting actually decreased later on in the experiment. And then we see this interesting increase around day 25, which is clearly not due to irrigation. We think this may be phenologically driven. So around this time, switchgrass is actually switching from the stem elongation phase over to earlier flowering. Here are the moderate drying plots, and fixation is on the same scale again. And what you can see is that the 30 millimeter rain events, which are double the previous ones every 10 days, had a more consistent response and really increased fixation here. And that as soils are drying down, we see a decrease in fixation. But there's still a decent amount of variation, as you can see here. Here are the extended drying plots. They receive 60 millimeters of water, as marked by that large red arrow on day 20. And what's interesting here is that fixation is increasing in response to wetting. But in half the plots, there's about a delay, so you don't actually see them peak until day 22. And then some of them stay even elevated at day 25. As the soils start to dry down, we see less fixation. So we're still seeing a lot of variability here. So what can we say conclusively when we look at all the data? Well, we know that if we compare fixation before and after wetting, there was a large increase due to wetting. But there's a lot of variation in that response. And so we wanted to know what influenced that response. So we used mixed model selection, where we looked at potential factors, including types of wetting or moisture, as well as we looked at nitrogen availability in the soil and in the irrigation water. And we were concerned about that is because if there's a lot of nitrogen that's abundant, we know that fixes are less likely to fix because it's not energetically profitable to do so. And then obviously carbon mineralization is representative of the energy available in the system. So what we found is that actually, of all the things we measured, the only 2 things that really influenced the response to wetting were the irrigation parameters. So how much water we added, which increased the amount of fixation, and then the amount of nitrogen in the irrigation water, which decreased the amount of fixation happening. And the interaction here actually means that as the plots are receiving larger amounts of water, that nitrogen present had a disproportionately large effect at those sort of bigger events. So there is that corresponding decrease there. And even though the nitrogen we added via irrigation was similar to what would have been expected from atmospheric deposition for this time period, the sensitivity of this energetically expensive process to N is striking. It can also mean that rain events in industrialized areas or those receiving high levels of N deposition may be less likely to stimulate nitrogen fixation. So something to consider for the future. Finally, when we examine the soil cores collected after the final wetting event from day 20 through day 30 as soils were drying down, we find that day explains the majority of the variation. Nitrogen fixation rates were higher closer to wetting and then decreased as soils dried down. But there was also variation due to rainfall frequency treatment as well. So here are all the post-experiment nitrogen fixation rates on a log axis, and then the rainfall frequency treatments here on the x-axis. And what we find is that the more extreme treatment, extended drying, had the highest fixation rates at the end of the experiment. Now, to wrap up what we've learned, we still observed a whole lot of variability in fixation temporally and spatially, but we're getting better at predicting the episodes. The remote control is now coming into view slowly, and at the very least, we know we should be tuned to the Weather Channel, because wetting events do increase fixation. The magnitude of the wetting event and its nitrogen content, though, is also important. We also want to know how rainfall frequency affects nitrogen fixation in different parts of the growing season, and that's something that we'll look at in the future. Not only is it important to consider how changing precipitation patterns could alter future nitrogen fixation rates, but locating this remote control for this process is really critical for the development of more sustainable biofuels. With that, I would like to acknowledge team members Matt Bellinger and Jabarius Jones for the help in the field and lab. Kevin Kmark helped us build the rainfall shelters. We couldn't have done it without him. And Hassan analyzed our stable isotope samples. So thanks to Hassan Gandhi. We are so grateful for everyone else who is listed here. And if you have any questions, please contact me at carmela.vita@wsu.edu. Thank you.